Good morning, everybody. Good morning, and welcome to yet another super cool, super awesome, and absolutely amazing session on the chapter Excretory Products and Their Elimination, lecture number 3. And today we are going to discuss about urine formation and the physiology of the kidney, sharp at 11:30. Yes, I hope you all can hear me and see the screen properly. Yes. And those who are watching the session, don't forget to click the like button right now on your screens. Moving on with the third lecture of the chapter, and we have two more lectures to complete with this chapter. Yes, Shraddha, thank you so much. Guys, I recorded. Soon after this lecture, you will have your results on our YouTube channel. And I'll share the link in our Telegram group. Okay, all the results, and you all made me so proud with your results because this is the best ever results that my students have given me. Neet mock test number 3. All of you made me so proud with your results and so happy for that. So happy for that. So happy. The best ever results you have given to me. Okay. So happy. Everyone is going good. So the third lecture, today we are discussing on the topic urine formation and the physiology of the human kidney. Physiology of the kidney as well as the urine formation. So these are the things which we are discussing in today's lecture. Okay, yesterday we lagged a whole day with our portion. So I don't want it to happen today. Yes. So there are mainly three main processes which are involved in the urine formation. There are mainly three main processes which are involved in the urine formation. First one is glomerular filtration. Second one is reabsorption. Third one is tubular secretion. So glomerular filtration, reabsorption and tubular secretion are the three main processes which are involved in the process of formation of urine. So we are moving on in detail with each and everything, and firstly we have the glomerular filtration. We studied in the last class what do you mean by a glomerular filtrate, or what do you mean by a glomerulus. This is a glomerulus, that is glomerulus we studied in the last class. It is a tuft of blood capillary. Glomerulus is a tuft of blood capillary. Yes, this is the thing we studied in the last class. Glomerulus is a tuft of blood capillary which have two main parts, that is the afferent arteriole entering into the Bowman's capsule and the efferent arteriole which is leaving the Bowman's capsule. Guys, can you all hear me? Are you all getting the point? Give me hearts on the screen. Give me hearts on the screen if you are getting out the points. Fast. Yes. Thank you so much. So glomerular capillary, what does this filtration occur? Here in glomerulus, what is mainly happening? The filtration of our urine takes place. Okay, there are certain glucose or whatever, sugar and everything may be present in our urine, and they all may be filtered out. So there is the filtration taking place where it is in the glomerulus. So why is the filtration taking place inside the glomerulus only? That is the question. This is a previous year MCQ. This is because there is a high blood pressure. There is a high blood pressure inside the glomerulus. So inside the glomerulus, due to the different diameters, in the diagram itself it is clearly shown. Due to the different diameters of the afferent and efferent arteriole, the blood pressure is very high. The blood pressure is very high because the diameter of the afferent arteriole and the efferent arteriole is very large. Different is there. So that there is a large blood pressure, there is a large difference in the pressure happening inside the glomerulus, which leads to the filtration of the blood. Okay, so mainly we have three main layers in the glomerulus. First one is the endothelium. First one is the endothelium layer, which is made up of glomerular blood vessel. Second one is the epithelium layer, which is of the Bowman's capsule. And the third one is the basement membrane, which is present between the two layers, clearly shown in the diagram. Yes, epithelium, outermost layer of the glomerulus. Next we have the endothelium, which is present inside. This is the endothelium, this is the epidermis. Next we have the basement membrane. Next we have the basement membrane. Guys, are you all getting me? Please respond in the chat box, otherwise I will be thinking that you are not understanding it. If you are understanding, just simply comment me yes or no. Yes. So the glomerular filtration is occurring due to the large difference in the diameter of the afferent and the efferent arteriole. We have three main layers in the glomerulus, that is the endothelium, epithelium, and the basement membrane. So all these things are the different layers of the glomerulus. Next, we have the epithelial layer. The epithelial layer contains specialized cells called podocytes. This is at least 25 times repeated MCQ. At least 25 times repeated MCQ question. That is the epithelial cells, or the cells which helps in the glomerular filtration. It is the podocytes. So podocytes of the Bowman's capsule are arranged in which type of manner? Are arranged in what? Intricate manner. Okay, sorry. It is arranged in an intricate manner, and how it is leaving some minute spaces called filtration slits or slit pore. Okay, this is the podocyte. So the podocytes, or the epithelial cells of the Bowman's capsule, are arranged in an intricate manner, leaving some minute spaces in between called the filtration slits or the slit pores. For what? For the filtration to happen, there is a filtration slits should be there, right? So this is the filtration slits, this curved portion. It is the filtration slit or the slit pore. So it is where the filtration is going to happen, and the podocytes or the epithelial cells are present in all these regions. Okay, take a screenshot for this, everyone. Yes, done, everybody. Yes, so this curved portion, you will call it as a filtration slits. Then this cells, epithelial cells, you will call it as the podocyte. And this is the podocyte, the cell body. Fine. Just recall the name only, that is the epithelial cells which helps in glomerular filtration, or the cells present in the glomerulus or the Bowman's capsule that helps in filtration. You will call it as the podocytes. Next one is the almost all the constituents of blood plasma except the protein. Almost all the constituents of blood plasma except the proteins passes into the lumen of the Bowman's capsule. Guys, don't miss the evening session. Don't miss this session at 4:45, because it's a very important session. Most of the students are feeling confused about the counter current mechanism. The counter current mechanism, most of the students are feeling difficulty, so all have to attend the lecture in the evening. So all the constituents of the blood plasma except the proteins pass into the lumen of the Bowman's capsule. What is Bowman's capsule? Bowman's capsule is the cup-shaped portion. Bowman's capsule is the cup-shaped portion of the nephron. So about 1100 to 1200 ml of blood is filtered by the kidney per minute. Very important question. This is an MCQ question. How much amount of blood is filtered by the kidney each minute or per minute? It is 1100 to 1200 ml of blood. It constitutes about one-fifth of the total blood pumped by each ventricles in a minute. Okay, one-fifth of the total blood pumped by the ventricles per minute. They are just filtering. The kidneys are filtering nearly about 1100 to 1200 ml of blood every minute, and it is constituting about 1/5 of the total blood which is filtered or pumped by the ventricles. Okay guys, have a look at this diagram, everybody. All of you have a look at this diagram. This is the afferent arteriole which is entering into the Bowman's capsule, and this is the efferent arteriole which is leaving the Bowman's capsule. This is the efferent arteriole which is leaving the Bowman's capsule. Okay, so guys, after the glomerulus, after the blood passes into the glomerulus, the necessary materials are passed on except the plasma proteins. All the other materials present in the glomerular blood just diffuses by the process of diffusion. By the process of diffusion, all these materials passes into the lumen of the Bowman's capsule. And you know that the Bowman's capsule is a structure which is going to get extended as the proximal convoluted tubule, loop of Henle, and finally into the distal convoluted tubule. So each part has different function, right? So all these materials are entering into the lumen of the Bowman's capsule by simple diffusion except the plasma proteins. Okay, so that filtrate which is obtained in the Bowman's capsule, we will call it as a glomerular ultrafiltrate, or this glomerular filtration, the process you will call it as ultrafiltration. Okay, glomerular filtration, another name for glomerular filtration is ultrafiltration. Okay, ultrafiltration on that material or the filtrate you obtained, you will call it as the glomerular filtrate or the glomerular ultrafiltrate. Next one guys, next one. We have 180 liters of the glomerular filtrate is produced daily, and about 99% of the glomerular filtrate is reabsorbed by the renal tubule. By the renal tubule itself, more than 99% of the total 180 liters of glomerular filtrate is reabsorbed by the renal tubules. So normally the volume of urine released is 1.5 liter. So this was a previous year JIPMAR question. Previous year JIPMAR question. The volume of which among the following is the volume of urine released by a normal person? What is the normal person releasing content of the urine? It is 1.5 liters. But 180 liters of glomerular filtrate is produced, but 99% is reabsorbed back. So the volume of urine released is 1.5 liters. And they will be asking, what are the components of the glomerular filtrate? What are the main components of the glomerular filtrate? That's an important question. So you have to say that the glomerular filtrate consists of glucose, amino acids, sodium ions, etc., which are reabsorbed actively, and the nitrogenous wastes are absorbed passively. All the filtrate components which are essential, that is the glucose, amino acid, sodium, all these things are actively absorbed. They are actively reabsorbed into the body, whereas nitrogenous waste such as ammonia, urea, uric acid, respectively in different organisms, they are absorbed passively. What is the significance of passive reabsorption? Passive reabsorption of water occurs in the initial segments of the nephron. What is the initial segments of the nephron known as? Bowman's capsule, PCT, etc. Okay, PCT, Bowman's capsules are the initial segments. Okay, so look at the part guys. You have a detail about this particular diagram in case when you come to the counter current mechanism. Counter current mechanism. So in the last class I told you the proximal convoluted tubule will extend downwards as a long tube, hairpin shaped structure, and that structure you call it as what? This structure you call it as what? Loop of Henle or Henle's loop. That structure you call it as a Henle's loop or the loop of Henle. And the efferent arteriole, this is the efferent arteriole, guys, right? So the efferent arteriole will forms a cluster. The efferent arteriole will forms a cluster with these. That is a Henle's loop, and it will form a tuft of blood capillary. It will form a tuft of blood capillaries like this. Okay, and it will form a tuft of blood capillary. Okay, so the passive reabsorption takes place in the initial segments, and guys, this blood capillary, I told you the name, that term itself, peritubular capillary, or you will call it as the vasa recta. Peritubular capillary or the vasa recta. This is what we studied in the last class, vasa recta or peritubular capillary, and we are going to study about each thing in detail in the upcoming class, that is in the evening session mainly. So this is what we studied. In selective reabsorption, sodium, amino acid, glucose are reabsorbed from the filtrate back into the blood, because the blood have to supply sodium to the necessary parts. For example, for creating impulses, sodium is required, so transported to the nervous system. Then amino acid, for energy production, it is required. Glucose for providing energy inside our body. So different materials are required in different places. So sodium, amino acid, glucose, substances like these which are actively reabsorbed into the blood filtrate. From the filtrate, these are absorbed back into the blood. Okay. So the tuft of blood capillary or the peritubular capillary, which is happening inside or occurring inside what? It is bound to see with the loop of Henle, you will call it as what? The vasa recta. Again, repeating it, vasa recta. I hope you are remembering. Then give me thumbs up on the chat box. Yes. Thank you so much. Peritubular capillary or the vasa recta. The proximal convoluted tubule reabsorbs most of the nutrients. You have to study this. Most of the nutrients are reabsorbed in the proximal convoluted tubule. 70 to 80% of electrolytes and water are also reabsorbed inside the what? Inside the proximal convoluted tubule, that is this region. This region you will have the most of the nutrients, electrolytes and also water are reabsorbed inside this area. This is a repeated MCQ for your exam. Very important area for Neet. Which of the following substance is reabsorbed inside the proximal convoluted tubule? What you can say for that? It is nutrients, 70 to 80% of electrolytes and the water. And in the proximal convoluted tubule, I hope you studied in the chapter structural organization in animals. You studied the simple cuboidal brush bordered epithelium which are required over here. Cuboidal epithelium is the type of epithelium which occurs in the proximal convoluted tubule. I hope you study. Give me hearts on the screen. If you're remembering, give me hearts on the screen, guys. I hope you written the exam also for this, so you know all these things. Right? About the proximal convoluted tubule has cuboidal epithelium. Yes, you studied it. Next is loop of Henle. That is this region. This region. Can you all see the pointer of the mouse? 1 second. So guys, this region. I hope you could all see the laser pen on the screen. So guys, this region is the loop of Henle region. Okay. So in the loop of Henle region, what is going to happen, you know? In the loop of Henle region, it maintains a high osmolarity. It maintains a high osmolarity of the interstitial fluid. So as I already told you that kidney are also helps in the osmolarity maintenance or osmoregulations For the interstitial fluid, they have to look out with the what? Osmolarity of that particular thing, also the interstitial fluid. Then this is the dissenting lymph. You know that this is the dissenting lymph. This is the ascending lymph. Yes. So guys, this top portion, this is the ascending lymph. This decreasing portion, that is a descending lymph. Look, descending, ascending. So the descending limb of the loop of Henle, guys, in the evening lecture, I'll be asking you and you have to give me the answer by yourself. Okay, you have to give me the answer by yourself about this. The descending limb is permeable to water, but almost impermeable to electrolyte. So the descending limb of the loop of Henle is permeable to water, but it is almost impermeable to the electrolytes. And what is its importance? It concentrates the urine. So definitely I'll ask you about this. In the counter current mechanism, this is the simple logic which every student needs. Since you are confused with this, you are getting it confused about the counter current mechanism, in which you will get a +4 marks for your exam. So guys, study either the permeability and the impermeability of descending limb, or you study the permeability and the impermeability of the ascending limb, so that no type of confusion occurs. Okay, so descending limb is permeable to water. So this descending limb is permeable to water. That is, water can enter and water can leave, but it is impermeable to electrolyte. That is, electrolytes will never be able to enter inside the loop of Henle's descending limb, or it will be allowing it, the electrolytes present inside it, to leave from that. In the ascending lymph, the minimum reabsorption takes place because it is impermeable to water but allows the transport of electrolytes. So the filtrate gets diluted. So name the part of the loop of Henle which makes the urine concentrated. Descending lymph of loop of Henle. And name the part of the kidney or the nephron that makes the urine diluted. Ascending lymph of the loop of Henle. These both are previous year MCQ for at least 20-25 times. Fine? Yes. Guys, do you want me to take a motivational lecture for on any of a topic? Anyone, do you have any suggestion? Because within 2 days, I'm on its preparation. So within 2 days, I'll be coming out with a dropper strategy for NEET 2021. There are certain repeating students also in our group, right? Those who have written the NEET 2020 and is going to again write NEET 2021. So I have been coming out with a certain dropper strategy for them also, because they should not go with this 2021 normal batch student, because 2020 students have already completed class 11th and class 12th topics. So it's their time to get maximum involved in the practicing of MCQ. But what about 2021 students? They have to look out with their 12th portion, 11th portion within this 1 year very carefully. 2022 students, you have a much enough time. So maximum care to your 11th standard topic, complete your 11th standard topic and perfect, so that in your 12th standard you would not trouble. The 11th standard biophysics and chemistry will never trouble you. Okay, fine everybody. You have to just concentrate on 12th itself. No need to concentrate back into your 11th standard if you are making each and every topic pakka. Yes. So if you have any suggestion on any topic that you need me to speak about, you have to tell me. I'll just record it and will share it with you. Certain tips you needed for your preparation or anything, you can tell me right now, or else you can tell me personally or in the group of Biopoint. Yes. Next one, in the distal convoluted tubule, conditional reabsorption of sodium and water takes place. So this is again a previous year question, a previous year MCQ. In distal convoluted tubule, that is a portion which is further from the Bowman's capsule, conditional reabsorption of sodium and water takes place. The collecting duct extends from cortex to inner parts of the medulla. Cortex is the outer region, and the collecting duct, I told you, collecting duct is the only portion which is going to extend into the medulla region. So it reabsorbs a large amount of water to concentrate the urine. It reabsorbs a large amount of water to concentrate the urine, and it also allows a passage of small amounts of urea into the medullary interstitium. What is medullary interstitium? The spaces, the interstitial spaces in between the medulla, to keep up the osmolarity. What is osmolarity? The balance between the water and the mineral balance of our body. Okay. Yes, we have 2 more slides to complete now. Next is the tubular secretion, which is the third step. So guys, can any one of you recall what are the 3 steps of urine formation? First one is glomerular filtration. Second one is tubular reabsorption. Third one is tubular secretion. Sorry guys, second one is selective reabsorption, you have to say. It's not, it is taking place inside the tube, but the actual biological term you have to use there is a selective reabsorption. Okay, last procedure of the urine formation inside our body, it is a tubular secretion. So the cells of the proximal convoluted tubule and the distal convoluted tubule maintain an ionic balance, that is sodium potassium balance is very essential, which we will study in the evening lecture. In the counter current mechanism evening lecture, we will talk about the sodium potassium balance and the acid base balance, that is a pH balance of the body fluids, which is by selective secretion of H plus, K plus, NH3 plus into the filtrate of the body and absorption of the bicarbonate ions from it. Okay, so the collecting duct is the part which maintains the pH and the ionic balance of the body by the secretion of certain, the term itself suggests tubular secretion, so they are secreting certain ions into the tubes. So guys, no need of further explanation, just take a screenshot for this. So guys, here, from here, the blood, all the protein except the plasma protein, everything goes into the Bowman's capsule. From the Bowman's capsule, electrolytes, water and the nutrients are absorbed in the proximal convoluted tubule, and the dissenting lymph is permeable to water and impermeable to electrolytes, and the ascending lymph again opposite to the dissenting lymph. Then it comes to the distal convoluted tubule, and finally to the collecting duct. The collecting duct, what you will have to see, the collecting duct, majority of water is also reabsorbed into the blood in order to maintain the osmolarity of the blood and also to maintain the urine concentrated. So guys, in the evening class, we will again see you back. So till then we meet. Bye bye. Take care. Okay. See you in the evening lecture with this particular thing, counter current mechanism. This is a topic which we are going to discuss in the evening lecture. So bye bye guys. Take care. See you sharp at 4:45 today. And after this lecture, you will have your result publications on your YouTube. Okay. Stay tuned.